Hello guys, today I'm going to talk about the role of the kidney in acid-base balance. So the kidney is really one of the most important organs that is involved in acid-base balance, and this is the utilization of three different buffer systems. One is the bicarbonate buffer, one is the ammonia buffer, and then one is the phosphate buffer. I'll be speaking about the ammonia and phosphate buffer because that's what the root of the kidney excretion is. Okay. So coming back to the ammonia. So from where does ammonia come from? It comes from an amino acid called glutamine. Where does glutamine come from? It comes from the liver and the muscle. Glutamine is converted into glutamate by glutaminase, and this releases a molecule of ammonia, one molecule of ammonia. The second one is glutamate is converted to alpha ketoglutarate through glutaminase, again releases another molecule of ammonia. However, this ammonia is trapped by combining with chloride in the medulla to prevent wastage of ammonia, as I would say. Okay, this is a very important factor to note that glutamase is a highly regulated enzyme and it is inhibited by hyperkalemia. So if you have high potassium ions outside the cell, it will really inhibit the glutaminase enzyme, and therefore we have no ammonia being produced, which could be quite dangerous on a serious note. Okay, so the next buffer I'll be talking about is the phosphate buffer. Where does the phosphate buffer come from? It comes from your plasma, obviously phosphates. These phosphate buffers is also known to vary with diet. Like the more phosphates you consume, the better your buffer capacity is. So let's say so it's also known as a titratable buffer because you can titrate the amount of acid being used. Okay. One more thing I forgot to mention is, for instance, in kidney disease we have hyperkalemia. So do we have a good acid-base buffer present? No, because we have hyperkalemia, which inhibits glutaminase, therefore preventing ammonia synthesis. So the only buffer we would really rely on is on the phosphate intake. But however, if we consume a lot of phosphate, there's a big problem. It can cause hypocalcemia because the phosphate combines with calcium ions. Okay, that I will be talking in the later videos, so no need to worry about this right now. Okay, so the second part of the tubule that you need to be worried about is the distal tubule. The distal tubule, and also the distal tubule. And the distal tubule is composed of two parts. The first one is the alpha intercalated cells, and the second one is the principal cells. According to our focus to the topic, we'll be focused on alpha intercalated cells. So they secrete hydrogen ions through the hydrogen ATPase pump. Then it also secretes hydrogen ions in exchange of potassium. So for instance, if we have hypokalemia, we need to absorb more potassium ions. How will we absorb more potassium ions? By activation of this pump. So hydrogen will be excreted in return. Okay, so now let's come back to acidosis. Okay, so if we have increased acidosis, that means our body will regulate in order to release hydrogen ions. But how is the hydrogen ions released? These two pumps are activated by a hormone called aldosterone. And if you remember, in the previous video I spoke about how hydrogen ions causes hyperkalemia because it needs to be exchanged with potassium in order to maintain the pH of the blood. So this hyperkalemia basically releases aldosterone. One of the biggest stimulus for aldosterone release from the adrenal glands is hyperkalemia. Okay, so let's say we have metabolic acidosis. We have hyperkalemia. Hyperkalemia causes release of aldosterone. Release of aldosterone activates these pumps. Therefore, we tend to start to develop alkalosis. And then, after all, since this acid is released, it needs to be buffered. I'll tell you why it needs to be buffered, why it can't be excreted normally. Okay. So these hydrogen ATPase pumps basically stop working at when the pH of the urine touches 4.5. And if you know that our pH of the urine is normally around 6 to 6.5, And our if our hydrogen pump stopped working at 4.5, then we can't really secrete acid, as we know that the hydrogen ATPase pump at the end of the day is an enzyme, so we can't secrete acid. So how will we really prevent this horrible effect from happening? We have our buffers coming to the rescue. The ammonia buffer, it will release ammonia chloride, so you'll be forming ammonium chloride, and this ammonium chloride is secreted in the urine as ammonium chloride. Yeah, this prevents the urine from being too acidic, so it can prevent the ATPase enzyme from paralyzing or from denaturing, I would say. Another buffer, we would have the phosphate buffer, which would come to form phosphoric acid. So let's say HPO4 plus H plus equals to H2PO4. At the end, this phosphoric acid is excreted in the urine. Okay. Yeah, I think so. That's it. One more thing I would like to mention is that what's the significance of understanding, like for instance, if we have a metabolic disturbance of acidosis or alkalosis, we tend to measure the chlorine ions. Why do we need to really mention the chlorine ions? Okay. Or urinary anion gap. What is the urinary anion gap? The urinary anion gap is composed of sodium plus potassium minus chloride. So what is the significance of this? Okay. The urine anion gap is useful in determining whether the metabolic acidosis is due to the renal problem, specifically renal problem, or is due to exogenous problem, which is let's say Addison's disease, then acetylazamide, then spironolactone, then normal saline. So how do we determine this? Okay, so for example, there's a condition called renal tubular acidosis. Renal tubular acidosis is when the distal tubule is not working. The distal tubule is not working, so therefore we can't secrete chlorine ions as well. Or we can't secrete hydrogen ions as well. And since hydrogen ions can't be secreted, ammonia chloride can't be secreted. Ammonia chloride can't be secreted, can't form ammonium chloride, so you'll have low chlorine ions in your urine. However, if the metabolic acidosis, I'm talking about specifically non-anion gap metabolic acidosis, is relatively due to another cause, then we would have presence of chlorine ions because the acid is able to be neutralized by your buffers. Yeah, that's it. Thank you. If you have any questions, please ask.